This is the explanation of both the glue and the agar. So I want to remind you first about the size of molecules. Molecules can come as individual units called monomers, they can come as double units called dimers, or they can also exist in this structure here, which is a polymer. Glue is actually much like this. It's a polymer. It's spaghetti-like. Now, if you can imagine this is floating in solution, that these little, well, actually very long, these long strings will be crisscrossing each other inside of the liquid. And then we're going to see what happens when we add the borax to them. So we took a look at three other polymers. The first one was polyethylene, which is found in plastic. We also looked at sodium polyacrylate, which is found in diapers. We also took a look at fiber, which is found in some products that you can get at the store, but also found in bread and other plant products. So this is going to be taking a look at glue. Glue actually is a polymer. And if you can imagine this being the glue, and with our spaghetti-like types of strings kind of crisscrossing like this. So this is actually floating in the water, and these strands are moving in and out of one another. But what we did to this is we added some borax to this. And I'm going to represent the molecule of the sodium tetraborate as just a B for the borax, or for the boron. And what the boron does is this is going to link the chains together. So you have your long chains, these are the polymers, this is the glue. So again, here's the glue chains, and you have these borax molecules which are going to link the chains together. So the borax, when it's mixed in with the glue, is going to tie those glue chains together. It ties those glue chains together. It ties the glue chains together. And so where these crisscross, it's going to link those chains together. And when we get these links, what's going to happen here is, now that they're tied together, this is going to make it more solid. It makes it more immobile since you're cross-linking all these chains. So since it's making it more immobile, that means it's going to become more solid-like. So you have all these crisscrossing chains, you throw the borax in, the borax then actually prevents them from moving anymore, and so therefore the chains can't move in and out of one another. It fixes them in place, and that creates more of a solid. It's not completely solid, it creates a semi-solid. You have both liquid and solid phases both at the same time. So that's the explanation of the glue. Let's take a look at what's happening with the seaweed, with the agar. So again, your agar is a seaweed product, and it comes from red seaweeds. And think of it as a juice. You can grind up the seaweed, mix it with water, you can make a juice, you could then purify these molecules. And this red seaweed juice is made out of sugars, very much like glucose. Glucose is represented as a hexagon like this. This is sort of like your C6H12O6. So this is like your sugar. This is just like the sugar that's found in your fiber, the glucose. But instead of a long chain of glucoses, it's a different name. So again, these are thousands of chains long, or thousands of sugars long. And what that does is it makes a long chain. So here's your chain of your thousands of sugars. So here's your individual sugar. But your chains for this are fairly linear, meaning that they're straight, like this. And when you heat this up in the liquid, these straight chains, they sort of rotate and move around like this because we're giving them lots of energy. So inside of our flask, draw a flask right here. Here's the flask. And this had the agar on the inside of it, and here are those chains. Now you have to remember these are microscopic chains. And when it's in liquid form like this, and it's being heated, these things are rotating because they have energy. So here it's a hot liquid. Again, this occurs with your pectins found in your fruit products. This also occurs with gelatin as well. These molecules are very, very long, and they're moving inside of the liquid when you heat them. So each of these molecules has enough energy to move. So these guys are rotating with energy. So they rotate with this energy. But then what happens is you could then cool this, and when you cool this hot liquid down, these molecules are going to start to lose this energy which allows them to rotate. And what happens is these molecules, they lay down on top in these layers. It looks, something like this. So when it's hot, it's a liquid and it rotates. When it's cold, these molecules, they lose their energy and they actually lay down flat in these layers. So this is when it's cold, and when it's cold, it becomes a solid. When it's hot, it's a liquid. When it's cold, it becomes a solid. And it also captures layers of water in between the molecules. So there's some water that's in between there. This is a perfect product for culturing bacteria, and that's what they do in a lot of microbiology. If you can imagine, here's your Petri plate. I'm drawing a top view of a Petri plate. So here's the top view. If you have a side view of a Petri plate, your Petri plate looks like this. It's a circular dish. You might have seen them in biology, and it might have this flat layer of the solid agar. And what you can do is you can take bacterial samples. There are many people that have jobs whose sole purpose is to take what are called swabs of the agriculture industry, for example. And what they'll do is they'll go into some type of plant and production plant or packing facility or anything that deals with food, and what they'll do is they'll take something that looks like a Q-tip, it'll be sterile, then they will swab this or swipe it across the countertop or whatever surface, then they'll come back to this agar plate, and what they'll do is then they'll zigzag this back and forth across the agar. So here's your Petri plate, and what's going to happen is the agar not only contains water, but it will contain food. And if there's any bacteria that was on the tip of that giant Q-tip, the bacteria would have been transferred now to this agar, and the bacteria now have a food source, and they also have a nice water source as well. So this is used a lot in microbiology for a lot of experimentation, for finding mutations, for finding new products in bacteria, doing DNA experimentation as well. So agar is used quite often in the industry of microbiology. Also, this is found in gel foods. Take a look at the food product that you're eating, and it might say agar as one of the products. So if it's a jelly-like substance, like an artificial donut that you might find in the store, your gel food might actually have agar, which comes from the red seaweed. And again, these are long chain polymers, and when they cool down, they will form these flat layers like this, and it will solidify into a semi-solid.